Usually when you look at a boring pile of landscaping mulch, you just see, well, dead wood. But what if I told you that right beneath your feet, there's like a microscopic Jurassic Park happening?
Yeah, a completely invisible and honestly absolutely ruthless one.
Right. So welcome to the deep dive. Today, we are looking at a stack of biochemical journals and field guides,
Hmm.
including a monograph called the Boreal Apex, to figure out why this incredibly ordinary-looking woodchip mushroom is secretly a microscopic predator.
And not just a predator, right, but one holding untapped pharmaceutical secrets. I mean, we are talking about Leratiomyces magnivelaris.
Yeah, that's the one. And this mushroom story actually starts way back in 1899.
Oh, right, on the Harriman, Alaska expedition.
Exactly. You had this massive ship full of scientists cruising the Alaskan frontier. And the eminent American mycologist Charles Horton Peck spots this fungus thriving in the subarctic tundra.
Which, is an incredibly harsh, nutrient-poor environment, and visually, it's pretty unassuming.
Right, it's just got this pale grayish-yellow cap.
Yeah, pale cap, dark purplish brown spores, and a thick, persistent membranous ring on the stem. But looking through the field guides, the real identifier is actually what it doesn't have.
Yeah, I was looking at the notes on this. It says this mushroom is missing something its cousins all have, which are these cells called chrysocystidia.
Yeah.
Why does lacking those even matter?
Yeah, so chrysocystidia are basically specialized cells that bounce light around like tiny prisms under a microscope.
Oh, okay.
Yeah, and most mushrooms use them to store waste or deter pests. But in this species, they are completely absent. I mean, even in its button stage.
Wait, so if it's trapped in frozen nutrient-dead soil and it's missing standard fungal hardware, how is it getting the energy to grow these robust mushrooms?
Well,
Because it has to be stealing it from somewhere else.
exactly. It doesn't just eat wood, it actually hunts.
It hunts.
Yeah. To survive and get nitrogen, it evolved specialized weaponry on its roots. The vegetative hyphae, it grows these microscopic, jagged spears in the soil called acanthocytes.
So they essentially lay down a literal bed of spikes underground.
A highly toxic bed of spikes, yeah. And it's a brutal four-step hunting process. So first, a tiny soil worm, a nematode like Panagrellus redivivus, bumps into an acanthocyte.
And then what?
Within minutes, the worm is completely paralyzed.
It's just horrifying. Does it, like, digest them alive?
Pretty much, yeah. Once paralyzed, those sharp projections use mechanical force to physically pierce the nematode's tough outer shell.
Ew.
I know. Then the fungus pumps in an enzymatic cocktail that dissolves the worm from the inside out.
Wait, so it paralyzes the worm, spikes it, and dissolves it inside its own shell. That's like trapping it in a microscopic Iron Maiden and then
Yeah.
just using the nutrients as fertilizer.
That is a spot-on way to picture it, actually. And then within 24 to 48 hours, the fungal roots absorb the nitrogen-rich remains.
Wow.
Right, that's the high-octane fuel pushing up those large mushrooms you see above ground.
Okay, so it's a worm killer. But the journals tie these predatory chemical weapons to modern drug discovery. So how does a worm-killing venom translate to shrinking cancer cells?
Well, the connection lies in how these chemicals work mechanically. To paralyze those worms, the fungus produces compounds like cytochalasans.
Right? Cytochalasans.
Yeah, and these actually target the nematode's cell skeleton. It basically locks its muscles in place so it can't thrash away. They also produce azaphilone alkaloids.
Okay, but how does that relate to cancer?
So, because these compounds are so good at disrupting cell structures, scientists tested extracts from related fungi on human cancer cells. By locking up the cancer cell's internal skeletons the same way, they actually dropped breast cancer cell viability down to just 28% in lab trials.
Wait, 28%?
Yes.
But wait, I have to push back here, because the sources say if you grow this fungus in a standard lab dish, nothing happens.
Right.
Like, it doesn't make these chemicals at all. So are we really on the verge of finding life-saving medicines in park mulch? Or is this just a quirk of the lab?
No, that's a very fair question. In a standard safe lab dish, the fungus has no predators and plenty of food, so its DNA keeps the recipes for these weapons locked away in what we call silent or cryptic genes.
Like, it doesn't bother baking the cake unless it has to.
Precisely. It needs a threat. When scientists co-culture it, which means forcing it to grow right next to a rival bacteria like streptomyces, it simulates the warfare of the soil.
Oh, so it panics.
Exactly. The fungus panics. Those silent genes wake up and it starts pumping out these novel, cell-destroying metabolites we've never seen before.
That is incredible.
Yeah, the potential here is massive because we finally know how to trigger the factory.
So really, the next time you walk past a damp pile of wood chips, remember you aren't just looking at dead landscaping, you are walking past a microscopic war zone, and quite possibly a future pharmaceutical factory.
It really is an absolute battleground down there.
It is, and it leaves you with something to cheer on. If a simple wood chip mushroom relies on bacterial turf wars to wake up its most powerful chemical defenses, what other life-saving silent genes are hiding in the everyday weeds and fungi around you, just waiting for the exact right trigger?